Welcome back, dear viewers. In this video, I would like to explain about the measles virus, which belong to the Paramyxovirus family. Introduction. The family of Paramyxoviridae includes three genera. First one is the Paramyxovirus, second is the morbillivirus, and third is the pneumovirus. Human pathogens within the morbilliviruses include measles virus. And the paramyxoviruses include parainfluenza and mumps viruses. And the pneumoviruses include the respiratory syncytial viruses. A new group of highly pathogenic paramyxoviruses including two zoonosis causing viruses, Nipah virus and Hendra viruses, which are identified in 1998. General characteristics of measles. Measles is a highly contagious virus which is spread through respiratory droplets. It is also known as rubeola, which is one of the 5 childhood classic infection, along with the rubella, roseola, fifth disease, and chickenpox. Historically, measles was one of the most common and unpleasant viral infections with serious potential sequelae, including younger than 20 years of age who had experienced the rashes, high fever, cough, conjunctivitis, and coryza of measles. In the developing countries, it has the highest morbidity and mortality among all vaccine preventable illnesses. Measles is one of the most ubiquitous and persistent of human viruses. Its distribution is worldwide and it causes diseases in any climate and under any conditions, which provided enough susceptible human beings who are brought together to enable it to spread among the people. Structurally, the paramyxoviruses are relatively larger in size with a pleomorphic envelope of 156 to 300 nanometer in diameter. The inner helical nucleocapsid symmetry is of 18 nanometer in diameter, and the nucleocapsid consists of a negative sense single-stranded RNA associated with the nucleoprotein NP and a polymerase phosphoprotein P, large L protein. The L protein is the RNA polymerase and the P protein facilitate RNA synthesis. The NP protein helps maintaining the genomic structure. Replication of the paramyxoviruses is initiated by the binding of HN, H or G protein on the virion envelope to sialic acid on the cell surface glycolipids and glycoproteins. The measles virus can bind to the CD46 membrane cofactor protein MCP present on most cell types and also CD150 signaling lymphocyte activation molecule SLAM, which is expressed on activated T and B cells. The RNA polymerase is carried into the cell as a part of the nucleocapsid. Transcription, protein synthesis, and replication of the genome all occur in the host cell cytoplasm. The genome is transcribed into individual mRNA and a full-length positive sense RNA template. New genomes associated with the L, N, and NP proteins to form helical nucleocapsid, which associate with the M proteins on viral glycoprotein modified plasma membrane. The glycoproteins are synthesized and processed like cellular glycoproteins. Mature virion then bud from the host cell plasma membrane and exit without killing the cell. Regarding the pathogenesis, the incubation period of the measles infection varies from 7 to 13 days. Measles is known for its propensity to cause cell fusion, leading to the formation of giant cells. As a result, the virus can pass directly from cell to cell and escape antibody control. Inclusions occur most commonly in the cytoplasm and are composed of incomplete viral particles. Viruses production occurs with eventual cell lysis. Persistent infections without lysis can occur in certain cell types. After local replication of virus in epithelial cells of the respiratory tract, The virus infects monocytes and lymphocytes, and the virus is spread through the lymphatic system and by a cell-associated viremia. Virus gains entry through respiratory tract, multiplies in the epithelial lining, and then spread to lymph nodes, where another phase of replication occurs. Further spread to organs takes place and skin, brain and lungs which gets involved. Lesions produced are characterized by the presence of multinucleated giant cells with well-defined intranuclear and intracytoplasmic inclusions. It is now well established that the maculopapular rash of measles is mediated by immunopathological mechanism. Such rashes do not appear in immunologically compromised patients who develop pneumonia, and in case of adults the disease proves fatal. The immune response of the host is as follows. The high titers of IgG antibody and IgM antibody as well as secretory IgA are seen after primary infection with the measles virus. IgM and IgA disappear after some time, but IgG persists for lifelong and makes the individual immune to reinfection. Though a strong immune response is mounted by the body on getting infected with measles virus, the disease has got an immunosuppressive action. The clinical features of measles, which is one of the most important childhood infection, the disease manifests with the prodromal symptoms of fever and upper respiratory tract infection, marked with coryza, cough, and conjunctivitis. Early diagnosis can be made by detecting Koplik spots, which are red macules or ulcers with a bluish white center seen on the mucous membrane of the inside of cheek. Rashes appear on different parts of the body, starting from head, followed by chest, trunk, and then limbs. After a few days, they start fading, and then recovery is rapid and complete. Measles can cause severe and multiple complications in a large number of patients. Nearly 10 to 20% of patients may have the complications like otitis media, pneumonia, croup, blindness, bronchitis, encephalitis. After 2 days of prodromal illness, the typical mucous membranous lesions known as Koplik spots appear. They are seen most commonly on the buccal mucosa across from the molars, but they may appear on the other mucous membranes as well, including the conjunctiva and the vagina. The vesicular lesions, which last 24 to 48 hours, are usually small and are best described as grains of salt surrounded by a red halo. The other complications of measles include subacute sclerosing panencephalitis, which is a delayed complication of measles, and this occurs some 5 years after infection of primary infection in the central nervous system, which leads to a progressive loss of cerebral functions ending in spasticity, coma, and inevitable death within 1 year, which are the functions of this rare complication, which occurs in 7 people in a million. Pneumonia, which is a serious complication, accounts for 60% of the death caused by measles. Mortality is associated with the pneumonia is higher in the malnutrition and extremes of age. The laboratory diagnosis of measles virus, which may be isolated from blood, throat, conjunctiva, and urine of the infected patients, which can be grown in cell cultures. Measles virus grows very slowly in primary human kidney, monkey kidney, and human amnion cell cultures. The virus produces characteristic multinucleate giant cell formation after 7 to 10 days of incubation. Cultures can then be tested for heme adsorption with monkey erythrocytes. The confirmation can ideally be made by immunofluorescent staining using monoclonal antibody. Demonstration of giant cells. Rapid diagnosis can be achieved by direct method of a demonstration of giant cells, that is Warthin-Finkeldy cells in respiratory secretions of or affected tissues. Treatment for the viral infection is the extensive use of live attenuated vaccines, which have brought down the incidence of measles quite significantly throughout the world. The vaccine is prepared from many types of strain, especially Gerald Lin strain, which was provided as measles, mumps, rubella vaccine grown in chick embryo fibroblasts. A single dose protects for more than 10 years. Thank you for your kind listening. For any doubts, contact through comment. Thank you once again.